And we'll start with structures external to the cell wall. These are prokaryotic cell structures. Generally, when people learn about cell structure, they learn about eukaryotes. You can take it. People generally are less familiar with prokaryotic cell structures. So let me just start with them. And again, we're on a little journey from the outside of a prokaryotic cell going to the inside, and we'll stop with the cell wall. So the first outer layer as we travel towards the center of the cell is a gooey, slimy, globular layer called a glycocalyx. And not all bacteria make a glycocalyx, but some do, and when they do, it can be medically significant, right? So, for a couple of reasons. Some form what's called a capsule, and a capsule is a glycocalyx. So first of all, what about the name glycocalyx? What does glyco suggest? Sugar, yeah, carbohydrates, glycogen, yeah, glyco is the prefix that means sugar. Calyx, that generally means crust or surface, yeah? All right, so a sugar coat, I think, is the way a lot of people remember this well. So it's carbohydrate based. It can be tight or it can be loose. If it's tight, we call it a capsule. It goes around an individual bacterium. If it's loose, it's more like a chocolate chip cookie, and the chocolate chips are the bacteria, and the cookie part is the glycocalyx. Does that make sense? That would be like a slime layer. You understand the difference? Right? A capsule, more like the sugar coat of an M&M. Yeah? Hard and individual. Each M&M is separate from the others. Yeah? But a glycocalyx can form something called a biofilm. And in the biofilm, your little lumps of chocolate are no longer M&Ms. They're more like chocolate chips in a chocolate chip cookie. Is that clear? And that's when you form a slime layer that forms a biofilm. Okay? You guys with me? All right. So, why do we care about these? Well, each of these is important. If a bacterium makes a capsule, that's important because it makes the bacteria more resistant to what's called phagocytosis. If you guys have heard of phagocytosis, then if you've got a capsule, it's harder to do that to a bacterium that has a capsule than one that doesn't. If you don't make a capsule, if you make a slime layer, that can make it harder to scrub you off of a surface. If you're bacteria that can land on the surface of your teeth, then you get tartar, biofilm on the surface of your teeth, yeah? Or plaque. Does that make sense? And you have to really scrub to brush it off. So, the glycocalyx can be essential to virulence. In other words, do you understand the term virulence? This is a term I haven't used before. It basically means deadliness, yeah? It means nastiness. In other words, ability to make you sick, right? So, as it turns out, Strep pyogenes has two strains, or excuse me, this is not pyogenes, this is Strep pneumo. So, Strep pneumo has two different kinds of strains, the R strain and the S strain. R is for rough, S is for smooth. And the reason for that is because when we look at colonies, can you see how these colonies look kind of goopy and slimy? You see these colonies on this plate? Right? These ones are goopier, yeah? And the reason that they are goopier is because this bacterium, Xanthomonas campestris, is making an exopolysaccharide, a glycocalyx made out of xanthan gum. Have you guys heard of this stuff? Where have you heard of xanthan gum? A lot of different things. What kinds of things? Foods, yeah. So The last time I saw xanthan gum, it was on the side in the ingredients list on a jar of salsa. Yeah? Isn't that nice? They're putting bacterial slime into your salsa. Why? Make it taste yummy. Yeah? So it's xanthan gum is carbohydrate-based exopolysaccharide made by Xanthomonas campestris. You grow it in a big vat, and then you grow the bacteria. The bacteria make this big slime coat. You scoop off the slime coat, get rid of the bacteria. Are there bacteria in your salsa? No, your salsa is generally pasteurized before you buy it from the jar. But it's got slime from bacteria to make it taste good. Isn't that nice? Is that bad? Are you guys grossed out? I'm partially saying this in order to gross you out. Do you guys eat yogurt? Yeah, so ideally, I'm trying to gross you out in order to tell you that you really don't need to be grossed out. Yeah? The fact that there's a bacterial byproduct is in your food is really not that big a deal. People, you can buy it. If you go to Bob's Red Mill, you can buy xanthan gum in a little bag and throw it into your chocolate chip cookies to make their texture nice or whatever. So it's a very common food ingredient. People buy it on their own to put it in their food. And if you're eating yogurt, you certainly don't need to worry about this stuff, yeah? Because in yogurt, you really are eating the live microbes. Here, you're just eating, right? You're committing genocide. Each little cup is like bacterial genocide every time you eat one of those little cups, yeah? Isn't it? Should we worry? No, they're bacteria, right? We should not care about... You're going to give yourself a headache if you care about bacteria living and dying, yeah? Although, you know, blessings upon you if you do care, right? You must be a very sensitive person if you're worried about the bacteria in your yogurt that are screaming and going, no, 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 please don't eat me. Right? Anyway, so this is why we're not making a lot of progress in class. Okay, so anyway, so here, bacterial glycocalyx, right? Delicious food additive. Here, little more medically relevant, right? 2 strains of strep pneumo, same species. One member of the species makes glycocalyx, makes a capsule in this case. The other doesn't. The rough strain doesn't, and when we look at colonies of the rough strain on an agar plate, they look all dry and crusty, yeah? And rough. We look at the S strain, the smooth strain, and the colonies on the agar plate look slimy and glossy, like this guy looks slimy and glossy, okay? And so it looks smooth. All right, we'll take some of these guys, put them into a syringe, and then inject them into a defenseless little mouse. What happens? The mouse lives, yeah? Take these guys, put them into a syringe and inject them into a mouse. What happens? The mouse dies. Why? Because, right, when you inject bacteria into the bloodstream of any animal, right, you're starting a battle to the death, yeah? Right? For the mouse, it's either that mouse or it's the bacteria, right? Same for the bacteria, right? It is a battle to the death when somebody does this, right? And the battle is between the bacteria and the mouse's immune system, yeah? Well, who wins against the rough strain? The mouse survives, and here the mouse dies, right? And the difference is the glycocalyx. These guys make a glycocalyx, and that's the difference that means the difference between life and death, both for the mouse and for the bacteria themselves, yeah? And the reason for that is because the mouse's immune system has white blood cells, and the white blood cells swallow up bacteria by phagocytosis, right? But if you have a slime coat, you're slipperier and you're slimier, and you're harder to wrap around for those white blood cells, yeah? And so the white blood cells aren't as effective in fighting the bacteria, and then the mouse dies, yeah? Does that make sense? Okay, so Glycocalyx matters, right, for health of mice and presumably health of people too, yeah? Okay. All right, so other structures. So much for the Glycocalyx. On to other structures. This is the flagellum. A flagellum, what does it mean? Well, the word means whip, but you can imagine what this thing is for. What's it for? It's for swimming, yeah? Bacteria swim in liquid media. Can you see how it works? How does the flagellum work? Does it wiggle back and forth? Look at the structure. See this disc here? This is called the basal body. See that? I guess the question is, does it work like A, a fish's tail flipping back and forth, B, a dolphin's tail moving up and down, C, a propeller like on a motorboat, or D, like jet propulsion, or E, like the oars on a rowboat? C is the correct answer. It works like the propeller. Does that make sense? Okay. By spinning around. And the way it spins around is actually, we'll see things spinning around like that in the future. All right. So you can have one flagellum. You can have many flagella all over your little bacterial body. You can have a few just on your tail. You can have two, one on each end, right? And how many flagella you have can help a doctor identify what kind of bacteria you are, okay? Is that clear? All right. If you have flagella, and not all bacteria do, then we say you are motile, which means what? You can move, right? And not every bacterium can move around on its own. Some just float around and expect to be buffeted by the tides, so to speak, right? So being able to move allows you to do what's called taxis. That is moving toward or away from a stimulus, yeah? Phototaxis, what does that mean? Towards light, or conceivably negative phototaxis, moving away from light, right? A worm would do negative phototaxis, would avoid the light. Cockroaches do negative phototaxis. They move away from light, yeah? Same with some bacteria. Some bacteria that can do photosynthesis would probably want to move toward light if they could move, yeah? Does that make sense? You guys with me? All right. What about chemotaxis? Moving toward or away from a chemical. Do you guys ever do this? You guys ever do chemotaxis? Give me an example when you do chemotaxis. Jump in a pool? What do you think? I would say chemotaxis is where, I hadn't thought of that. So chemotaxis, I think, is where you're walking down the street on a rainy day and you smell delicious food in a store, and you go into that store because you can smell the food, yeah? Or somebody rips out a thunder blast and you leave the room, right? Negative chemotaxis, yeah? Is that clear? All right. So bacteria can move toward or away from the chemical gradient in the same way that humans can move toward or away from smells, yeah? And smells are chemical gradients, right? They are chemicals going into your nose. What a lovely thought. Okay. Why do we care about flagella? Well, flagella, your body knows about. Your body, when you are born, right, much of your immune system has to develop as you grow. But some parts of your immune system are hardwired. And your body has evolved, your immune system has evolved to detect flagella. And the reason is because your cells don't have flagella. Yeah? Is that true? Are there any human cells that have flagella? Sperm cells have flagella, don't they? So, however, sperm cell flagella are different from bacterial flagella. And I'll explain that in more detail later on, okay? It's not the same. Your immune system, if you take a bunch of bacteria and you shake them violently and shake off all their tails, and then you centrifuge them to purify out their tails, and you have a little syringe filled with little bacterial flagella but no real bacteria, and you inject them into somebody, that person can go into shock. That person will have an immune response just from the tails themselves. Does that make sense? The tails are enough to tip off your immune system that there's an enemy around. Got it? Okay. In other words, flagellar proteins are antigenic. Your body will recognize them. Okay. Some other things. Fimbriae and pili. Fimbriae are little fibers that are not flagella. They're not for moving around. They're for attaching to things. Yeah? They're kind of like the fibers that allow mussels and mussels to sit on rocks, yeah, and barnacles, yeah? And then, oh dear, sorry, here's your R-rated picture for the day. See, this is really X-rated, isn't it? So, pili are another structure. These are hollow tubes through which DNA can sometimes be exchanged. Yeah, so this is two bacteria. How do bacteria reproduce? Not like that. They divide by binary fission, yeah? Right? That said, David is correct that this is bacterial getting it on, if you will, yeah? This is DNA exchange, yeah? But bacterial getting it on doesn't result in more bacteria, right? So this is kind of a strange concept, right? We associate genetic exchange with reproduction. You understand? When we have kids, our kids represent a genetic exchange between two parents. Does that make sense? And that person is the result of that genetic exchange, yeah? But here, there's genetic exchange. There's two bacteria that are, you know, they fell in love and got married first, of course. But they are doing genetic exchange. When this is done, how many bacteria will there be? There will be two bacteria, the same number that they started with, right? Well, what's the result? Well, one of them will be genetically different from the way they were before. Is that understood? You guys get that? So, this is genetic exchange, but this is not reproduction. How does reproduction happen? It happens by binary fission, right? So here, reproduction and genetic exchange in bacteria happen in completely different events. Does that make sense? And that's weird because they happen there's no way to separate them for people. Does that make sense? Okay. All right, so that's enough for now. That is structures outside the... Oh, wait. Why are bacterial capsules medically important? What's the answer? 4. It has to be 4. I talked extensively about 1 and 2, but it looks like 3 must also be true. If 1 and 2 are true, then it has to be all of the above. That means 3 is also true. You guys with me? Okay. So the fact that bacteria are in a matrix hanging out together means that they can send little chemical signals to each other and they can actually talk, and bacteria talking to each other. That was briefly mentioned by the lady Bonnie Bassler in that little movie we saw before our lab a couple weeks ago. But it's a pretty important concept. All right, so that